It is my pleasure to introduce Dr. Matthew Campen from the University of New Mexico. Dr. Campen is the director of the New Mexico Center for Metals in Biology and Medicine and the Regents Professor of Pharmaceutical Science. Dr. Campen's research on bioaccumulation of microplastics in human brains has revealed alarming findings. The study was published in Nature Medicine and highlights the presence of microplastics in human brain tissue, with concentrations higher in participants with documented dementia. Dr. Campen will be presenting Bioaccumulation of Microplastics in Descendant Human Brains. Dr. Campen, the floor is yours.
Thanks so much, Kevin. I appreciate the opportunity to share our work with HealthWatch USA, and I'll jump right in. This is going to be an overview of the paper that was published, as well as a lot of the background of the methods we use, the uncertainties involved in the data we've generated. And I just want to have opportunity for frank discussion about where we go from here and improvements for the research world. So starting financial disclosure, I'm a scientific advisor now for Circulate Health. It's just started, doesn't really enter into what we're doing here. The research funding for the work we've done comes from the NIH, National Institute of Aging, is the primary sponsor for this work. My sort of beginning into this world of plastics started with middle school science fair project with my kid. This is Sam. He's standing in the reservoir of Rio Grande, which is up here, very close to the headwaters, west of Creede, Colorado. And we sampled some water there. We worked our way down the watershed with increasing populations that contribute to the watershed. So you see the watershed carved out here in blue. We've sampled at Taos, and then we sampled down in south of Albuquerque, right next to the Isleto Pueblo. And what we found was maybe not surprising. Is you could look at this with a fluorescent light in standard light microscope and show that there were more plastics on the filter as you went down the Rio Grande, more human involvement, more contributions. But what was really surprising is pretty close to the headwaters of the Rio Grande in, this beautiful country up in southern Colorado. It was really easy to find plastics of all shapes and sizes. There were certainly things that looked like they came from fishing, but there was also small microplastic dots and all sorts of shapes and sizes. So this is the reality that we live in right now, is plastics have been produced over the last 80 to 100 years in exponentially increasing amounts. The amount we produce doubles about every 10 to 15 years. And there doesn't seem to be any restriction or abatement of this pattern. The problem is that once you throw it away, it starts to degrade, and that degradation can take years to decades. And as it degrades, it breaks into smaller and smaller pieces, and those become the microplastics. So that curve of increasing microplastics that's been measured on the surface of the ocean, shown here on right, that curve mirrors the generation, but it's delayed by 20, 30, 40 years because that's the time it takes. So the concerning part for me as a toxicologist is we don't necessarily know when these micro and nanoplastics are going to become bad for us. But what we do know is that we need to figure it out now in order to make an effect generations from now, 40, 50 years out, because that's how long it'll take for us to really stem the tide of this growth curve. The other thing worth pointing out is that there's now over a ton of plastic waste for every human on Earth, and very little of this is recycled. Most of it goes directly to landfill or is incinerated. Small amount is incinerated, but this little tiny curve on the bottom here, this is the recycled amount. 10% gets recycled, and about 1% gets recycled twice. Very little of it is really in this area. Most of it is just going to landfill or put in the oceans or, dumped somewhere. Small amount is about a quarter of it is still in use. So it's the coffee maker on your counter or the dashboard of your car. There's still a lot of that in use and more being produced all the time. Figure on the left depicts this process. You start with a macroplastic. It breaks down to microplastics. Microplastics are plastic particles in the range from 1 micrometer up to about 5 millimeters. The thing we're starting to worry about because of the observations of this study are the nanoplastics, which are less than 1 micrometer. And I like using this figure done by Peter Witch out of Australia, which gives you some nice correlation between that size. So at 1 nanometer, you're really in the range of molecules. We measure elemental size in angstrom, which is 1/10 of a nanometer. 10 nanometers is the size of big macromolecules like DNA. When we get to 100 nanometers to 400 nanometers, that's the size of some viruses. Cells in the body are going to be larger than 1 micrometer. Your nuclei in cells are about 5 micrometers. The biggest cells of the body, megakaryocytes in your bone marrow, will be around 60 micrometers. And then you get up to the things you can see, like grains of sand, and now you're talking about millimeter size again. But importantly, what we're seeing in the body that I'll show you as we get through this are in the size of 100 to 300 nanometers. So they're about the size of viruses. And really, the problem related to our lack of knowledge, our uncertainty around this issue stems from a lack of technology to accurately detect these and confidently detect these. So after doing some work with my kid, eventually he didn't even get a chance to present that because COVID hit and his science fair got shut down, which was really sad. But our group continued doing this research, and we started doing some studies with polystyrene microspheres, which are purpose-made for research, and they're very pristine and shiny and new. And we decided to test, well, what happens if they just get weathered a little bit? So we put it under a UV light for a month. We could see that physically degraded and chemically degraded the surface of these particles. And when we put them in a simple assay, this is just a cell culture assay where we wound the cells and we watch them regrow on the plate and how long it takes. We could show it with the fresh plastics. There was a small amount of inhibition of growth in terms of the rate of recovery. But when the plastics were aged, there was a significant, a really pronounced reduction in that recovery rate. And so that led us to realize, wait a minute, we just don't know what material we should even be using for toxicological studies. We know that these plastics are out there in the environment. We don't know that they're round. We know that they're mostly not polystyrene. They're more polyethylene, polypropylene, and other plastics. But we certainly know that their surface is going to be different because it's exposed to heat and water and UV light and all sorts of oxidants. So it led us to this moment. I don't know if you recognize this image from Time and Forrest Gump, where he stops and he has this moment of inflection saying you've got to put the past behind you before you can move on. And this is Monument Valley, which is up the road from where we live here by a couple 100 miles. But we knew we had to do something fundamentally different. We couldn't just apply these fake plastic particles to cell culture and expect to get anything really relevant without diving into understand what is actually in a human body. So this is sort of the journey we took. We started talking with our colleagues at the Office of the Medical Investigator about getting parts from cadavers, essentially. We knew that plastics were going to be hard to detect. They're essentially the same composition of human material. They're carbon, a lot of organic material, carbon, oxygen, and hydrogen. Most of the methods that have been used to date have been visual microscopy, and the limitation there is size. Most microscopes, especially with the types of detection methods, visual spectroscopy, there's Raman and FTR spectroscopy, those really can't image anything smaller than 5 micrometers. So the nanoscale stuff is just out as far as positive confident detections. So we felt like we needed to do something different. And we developed this path using a system called a pyrolysis gas chromatography mass spectrometer. And what this does is you isolate the plastics from the tissue and you combust them. And those plastics release sort of fingerprint gases that you can then look for specific chemicals and link that back to what you find from combusting pristine plastics at a known concentration. So it gives you a chance to identify as well as quantify the plastics. And I'll talk about some of the uncertainties, but it's important to note this is a very indirect method. We're looking at the combustion byproducts of polypropylene, and that ends up being a series of different molecules like 2,4,6 trimethylnonene, which is relatively unique and it's not a biologically derived thing, but It's not polypropylene, it's a piece of polypropylene. But this is a very useful method. And I liken it to this quote from Marcel Proust: "The voyage of real discovery consists not of just seeing new landscapes, but in having new eyes." And this was a new tool that really gave us an opportunity to explore the problem with a fundamentally different approach. It was quantifiable, it was cumulative. We're now able to capture all the nano-sized stuff, which I think is going to be really important. So the process here is we saponify the tissues. We use potassium hydroxide. We digest that for 3 to 5 days until it's thoroughly no longer a piece of organ, whether that was a brain or a liver or kidney. And then we spin it in an ultracentrifuge to isolate the solid particulates that are residual. There's additional steps that are embedded in this, but ultimately we take the pellet and we add it to a little stainless steel cup. In the pyrolysis GC mass spec, we incinerate that and we derive the spectra. So there we're able to quantify. And the first application of this was with human placentas in a collaboration with Shirsti Agard and Enrico Barroso at Baylor University. And it was really just fascinating look because we're able to show that there's a little bit of plastic in all of these placentas. Some of them are quite low, 6 micrograms per gram, barely detectable. But some placentas had quite a lot, over 600 to 700 micrograms per gram. And we were able to also identify different types of polymers. And this is a rough way of doing it, because in fact, if you think, a lot of plastics are really mixtures of polypropylene and polyethylene, or polyvinyl chloride and some other things. But this gives us a nice way of separating these out and showing, okay, most of what we're seeing is polyethylene, polyvinyl chloride, polypropylene, PET. SBR is styrene butadiene rubber, which is tire wear typically. So really fascinating way of doing this. We followed that up with a paper where we were able to get human testes from cadavers and compare those with dog testes from veterinary clinics in the region. And it was interesting to see that the human testes had 3 times the amount of plastics in them, but the overall distribution of plastics in both tissues was relatively similar. Again, polyethylene, polyvinyl chloride, nylon 6, nylon 66 being among the most prevalent. The other really interesting aspect of this was when we looked at the plastics in the human testes versus the age of the donor, the cadaver at their point of death, their time of death, it was not just linearly accumulating with age. I think there's this assumption that the plastics get in us and they don't get out and they just build up as we get older. That did not seem to be the case. And this graph in the lower right-hand corner is from another paper where they looked at the metabolism of testes over time or across age. And it shows a kind of a similar pattern that in peak reproductive years, this goes up. Metabolism of the testes goes up when you're actively generating sperm. And then as you get older, it starts to wane. And so, forest for the trees, I'm looking at this plot in the lower left-hand corner, and it's a bit of a Rorschach test. It's not statistically significant to link this to that kind of a curve, but that's what I see. It looks like it's higher in individuals from 20 to 50 years old, and then it seems to go down. So that brings us to the Nature Medicine paper. What we did here was we were able to obtain samples from the cadavers, again, from the Office of the Medical Investigator, and we applied 4 major methods of detecting plastics. They're complementary. They're not all quantitative. They all have some degree of uncertainty associated with them, and that's important to realize. The big thing was the pyrolysis GC mass spec. We also used Fourier transform infrared spectroscopy. We used a technique called polarization wave imaging, and that's what you see here on the right-hand side in the upper panel. You see the little tiny white dots that really pop out when you look at polarized light coming through them. That is not a chemical confirmation that it's plastic. It could be some other sort of crystal, and that's important to keep in mind for the uncertainty. But we wanted to do this because it gives us this anatomical information that we can't get from the homogenized sample. We also used transmission electron microscopy, again, to sort of visualize these plastics and the location within the tissue. So we were able to get 20 to 30 samples of liver, kidney, and brain from individuals who died in 2016. And the first thing that we saw was that, wow, there's ample plastics in every organ, but tenfold higher amount of plastics in the brain samples. We went back to the office of the medical investigator and said, you know what, this is really fascinating. These were the discarded samples from 5, 6 years ago that you were getting rid of. Can we get something more contemporary just to see if there's any kind of temporal relationship? So they got us freshly generated samples from 2024, and we were able to put the data together. And what we found was that, at least in the liver and in the brain, there was a significant increase in the quantity in 2024, suggesting that yes, this is something that's going up in our bodies over time, which is really not that big of a revelation if you consider if you got samples from 1910 or the 1900s or the 1800s, they would have no plastics in them. We didn't have plastics back then. So obviously there's a temporal component to this, and it's not too surprising to see the trend. The other issue that was fascinating was we were able to get samples from dementia cases. Those are shown in purple here. And then those dementia cases, they were 5 to 8-fold higher than the neurologically normal brain samples. These are all from the prefrontal cortex. The reviewers asked us to get samples from earlier time points, and that was not the easiest thing to do, but working with Andrew West at Duke, we did get a handful of samples from different clinics in Maryland, Massachusetts, and North Carolina. And ultimately, they were far lower when you go back to 1997 to 2013. So they're geographically different, but the time course, the regression curve that we draw through this data didn't change at all. There's still clearly a time component to this. So speaking about the uncertainty, the way we do this pyrolysis GC mass spec, as I mentioned, it's an indirect metric. Polyethylene in particular looks like fats when you do this. It's not exact. We've been testing just pure fat, sphingomyelin, cholesterol, things like that with pyrolysis GC, and it doesn't purely replicate the signal that polyethylene gives. But it's important to show this is our distribution plot on the right-hand side, and the orangish yellow bars show a large amount of polyethylene in our samples. That doesn't really reflect what we would find in the environment. We would expect to see a plurality of polyethylene, but certainly not a dominant majority. So we do think that our overall quantities are somewhat inflated because of the polyethylene. But it's important to consider that when we did our digestions to remove the fats as much as we could, we did this calculation called a digestion efficiency. And one would expect that if you didn't digest the fats away and you had less efficiency, you would have much higher amounts of polyethylene, and that wasn't the case. We really didn't see a relationship with that. The other thing is that each individual polymer has its own unique pyrolysate or molecule that we're measuring. And so just because polyethylene is vulnerable to this interference from biological matrix, polypropylene is not known to do that. Styrene butadiene rubber is not known to do that. And they're all showing a significant increase in our samples over time. So ultimately, we're still working on improving this method and making sure that it's really as high quality, high confidence as we can. But the concerns, there's been a lot of pushback, I would guess, from petrochemical industry. And I think some of it's justified. We're not there. This isn't the kind of data you would build a risk assessment on, but it is highly, it's highly alarming from the standpoint of these are clearly in the brain and it's getting worse over time. Thinking anatomically about this, we moved to this polarization wave imaging. As I mentioned, this is not chemically specific, but you can see what happens when you switch on the polarization wave filter. You can see these little particles that really pop. So these are immune cells from an inflamed region in a dementia brain. And you can see particles all over the place. In the kidney, it was interesting because the particles tended to be in the glomeruli, as well as along the distal tubule over on the right-hand side. In the liver, they really tended to be in lipid droplets. So you see these like haystacks of plastics piled in the little lipid droplets. Occasionally, you would see them scattered around amongst the hepatocytes. In neurologically normal brains, it was very hard to find plastic particles large enough to image. what we tended to see was these clusters of very, very nano size. And just for reference, you can see the nuclei here. Those are about 5 micrometers. So this is a larger cluster of greater than 1 micrometer, but all these other things are far less than 1 micrometer nanoscale. In dementia cases, it was really obvious, and they tended to cluster in two major areas. One is the inflamed regions of the neurodegenerative diseases, as well as along the blood-brain barrier. So if you look along the vascular wall, you would see large accumulations of these things. This is an important point to harp on. This is an associative study design. We have no evidence that the plastics cause dementia. We could just as easily conclude that the dementia disease creates a more inviting environment for the uptake of these plastics. It's known that dementia cases typically have a reduced blood-brain barrier. And so it could just be that these things are getting through in higher quantities. We're following up on this with animal models and trying to sort out what's first, the chicken or the egg. But at present, we really aren't making a strong statement about causality. The nice thing is other researchers are starting to dive in on different techniques to sort of buoy our initial findings with polarization wave imaging. So Elaine Barer is a neuropathologist at UNM, and she's using a sort of hyperspectral approach, and she sees these little, you see the little blue dots along these brains. This is also from a dementia case, and again, it's all along the blood-brain barrier. You can see the red blood cells are in the middle of this little arteriole, and there's just little dots everywhere. Another study out of China, these were mice that were purpose exposed to little polystyrene nanoparticles. And you could see not only are they getting into the brain, very, very clear purpose, cause, these are getting to the brain, but they tended to be in and around the blood vessels. You could see the transit almost from these still images. The other really wild observation from this study was this image. I think it's figure 2D. And this is what happens when we pull the plastics out in that pellet, and then we dissolve that pellet in an organic solvent and just spread out all the particles on a TEM grid, transmission electron microscopy grid, and then we visualize at a high magnification. So you can see all these little things that look like shards. And these shards are all universally less than about 400 nanometers. Most of them are down in the 200 nanometer range. So at this point, I'm going to step away from that Nature Medicine paper. I'm going to talk more about this article that's still in preprint. It hasn't been accepted for publication yet, but this gives you more information about what we were seeing with these shards. So for one thing, the Raman spectra of these shards was very consistent with fresh polyethylene, except that it had this additional peak from 1600 to 1700, which is a known region where highly oxidized polymers develop this carbonyl peak. So it's very consistent with a very old plastic. We heated these shards to the melting temperature of polyethylene, 130 degrees, and you can see this pseudo animation as I click through these slides. They melt and fuse together at that temperature. And then my good colleague Aaron Ederle, he might still be a scientist at NIOSH. It's unclear. Aaron worked with the team at RJ Lee in Pittsburgh to do manual sizing of over 3,000 particles from 3 different organs, from 5 different donors. And what he found, force for the trees, is that there's more large particles in brains. That might be part of the mass increase that we see in brains, is that there's just an accumulation of larger particles. And that was significant. So the length and the width are shown here in the bar graphs. And you can see that there's very little overlap between the brain data and the kidney and liver. But also just forest for the trees. These are very small. The mean length is less than 200 nanometers and the mean width is less than 50 nanometers. This is in the realm of the size of irises. We're not the only ones thinking about this. I had a talk with a University of Columbia colleague, Sanat Kumar, recently, and he says, yes, when I take fresh plastics with highly crystalline regions, so polyethylene, polypropylene, and I break them down, we start to see these nano shards too in this sort of 10 to 100 nanometer range. So I think we're really onto something with this. We got some pushback from reviewers saying they think we might have created these artificially, the way we digest. And so we went and talked to some colleagues over on our main campus, Arts and Sciences campus, And Joe Cook and Jonathan Dunham were able to get us some wild-caught rodent samples dating back to the 1960s and 70s. And this is what we saw in deer mice. So the Pyrolysis GC again showed us this temporal curve of increasing over time. The imaging from 1987 kidney samples, we really didn't see many particles, but they were still there. 2005, 2015, you start seeing more and more of these. We approached this with a more blinded study. Eve Rowland is a PhD student. She just defended. She's a real doctor now. But she utilized these samples of kangaroo rats and took the brains from kangaroo rats that had been in glass jars stored since 1966. She then went out to where they caught them in Rio Rancho, just north of here, and trapped some more mice or rats. And so we had a head-to-head comparison. And this is just it. This is Forest of the Trees. Here's 6 images from these samples. We have hundreds more of this, but 1966, you see things, but they are not these shards. You can sort of zoom around on these and, we look over and over, but in 2025, we have these shards. So are these from plastics? I'm saying they are. Other people are going to push back on that. They are definitely anthropogenic, I think is the final say on that. Why these are important is if they're this small, they really have a chance to be embedded within the system we use to uptake fats in our diet. So chylomicrons are the initial carrying system for the fats we take up through our gut. This is sort of a to scale artistic representation of what we might find, but these shards would easily fit within one of these chylomicrons. We did a quick study in mice, gavaging small nanoscale material, so 500 nanometer versus 1 micrometer, 1000 nanometer. What we found was that putting these into the guts of mice, we see in the blood, in the chylomicron fraction, they're very clearly translocating there, but we don't see that with the larger plastics. So we're following up on this in studies right now. We think this is really important because it tells us how we get exposed and how those particles may be distributed, because all these little proteins on the chylomicrons will help target it to tissues according to metabolism and need. And the implications for this are, if they're trapped within these chylomicrons, they're not obvious to the immune system. They will go to places that want fats for energy or fats for growth. They will be stored in regions with a lot of fat in them, and they will ultimately be able to evade immune recognition. So learning more about this is going to be really essential as we go forward. So I'll switch from there to talk about some controlled exposure studies, because the problem with the human data is it's all very associative. It's a cross-sectional study design. So this was a study we did where we had mice exposed orally to different mixtures of plastics, either polystyrene or a mixture of different polymers, for a 4-week period. And we then digested the samples and went in, and we were able to identify that, yes, we found the little plastic particles in anywhere we looked. They got to the brain, they got to the liver, they got to the kidney. This is not very quantitative, but it was proof of concept that, yes, these things are small and they get into the body. We then did some metabolomics to see if there were sort of major perturbations of function of those organs. And we looked in the colon, and we could see that basically the dark dots are lots of metabolites being altered as a result of this exposure. In the liver, you see again a lot of dark dots, but less. And then in the brain, it does seem to be that in this short-term study that the brain is less impacted, and maybe there's less translocating to the brain, but it's a short-term study. It's only one kind of particle, and we're trying to follow up on this with more realistic particles that resemble those shards. And another cool study, it's fascinating mostly for the overall concept. This is from Jamie Ross's group at the University of Rhode Island. And what she did was look at mice that had a knock-in humanized ApoE variant, either ApoE3, which is protective for Alzheimer's disease, or ApoE4, which is a risk factor for Alzheimer's and other dementias. The reason this is so interesting is that those chylomicrons have, as a carrier protein, ApoE on them. And so there could very well be some difference in how plastics are taken up and carried and transferred to the brain as a result of this. And what she showed, which was pretty fascinating, is that in a sex and genotype dependent manner, you did get alterations in behavior as a result of the plastics, and that the ApoE4 tended to be the one that showed the greatest amount of response. So very preliminary. Really, all of this is very preliminary. We're talking about a lot of data from one lab, new data from University of Rhode Island, but I think ultimately you need to keep in mind we need a lot of different labs jumping in on this with different perspectives, different methods, and really getting a sense for the forest of the trees. We've only been doing this work for less than 5 years. You think about how long it took for us to establish that cigarette smoke was bad for you, talking decades, and you had a really good control group, people that didn't smoke. We have a problem that everybody's exposed to these plastics, and so it's going to be really tough to sort things out. And I'm optimistic that more people are engaged and excited about this research, but it's going to take a lot of time and effort before we really sort things out. So, concluding thoughts, micro and nanoplastics are present in our bodies. We've shown this, a number of other labs have shown this. There are more papers coming out that will confirm what we've shown. We found that the levels in the brain were much higher than expected. They appear to be rising over time, and they were much higher in dementia samples. But the health impacts remain unclear. Everything here was an associative study. We are developing new methods for cerebrospinal fluid detection of plastics, and that's this graph up in the upper right corner. The importance here is that now you have an opportunity to take samples from individuals who might be at high risk for dementia. You could then put them on a specific protective therapy, diet, exercise regimen, whatever, and then look to see if there's improvement and then measure the amount of plastics before and after in the cerebrospinal fluid. So it's not an easy sample to collect, but it is something that you can do repeated measures with that, and it will be really important for our understanding of cause and effect. And then lastly, environmental nanoplastics are still what I would consider, and I know polymer chemists who would also consider them to be conjectural. Do they exist? Are they in high concentrations? Are they really degrading slower and accumulating, or are they degrading faster because of the high surface area? Lots of questions remain, and I think we need to get more data on these to better sort out how they're getting into us, how they might be accumulating in the environment and in our bodies. And I can stop there. I just like to highlight this is such multidisciplinary research, and a lot of people are involved, but Marcus Garcia has led so much of this. He's been a postdoc in my group. Hopefully we're getting him into a faculty position soon. And Eliana Hayek is a environmental chemist slash toxicologist. She's just inspired with the approaches that she applies to these questions. So I can stop there and take questions. I'll just say that if people are interested in hearing more about all this, we do have a meeting coming up in January that's going to be held here in Santa Fe, and we'd love to host people out. I think we've already got 90 abstracts submitted, so it's going to be a good sized group of people.